Welcome everyone to the virtual event on genetic testing for rare cancers, the importance of knowing your family history. This event is made possible by the Dan McMenamin Endowment and in partnership with the Weill Cornell Medicine Meyer Cancer Center Office of Community Outreach and Engagement. The Dan McMenamin Endowment is dedicated to the life and legacy of a Weill Cornell graduate, Dr. Daniel T. McMenamin. The endowment seeks to improve care, coordination, and collaboration for patients with rare diseases by increasing awareness, improving access, and fostering cooperation among patients, physicians, and researchers. We want to thank the family and friends of Dr. McMenamin for their support and contribution to this program. My name is Antonio De Rosa, and I'll be your guide this evening. A little bit about me. I'm a faculty member at Weill Cornell, serving as the program lead for decision navigation and patient support in the Meyer Cancer Center's Office of Community Outreach and Engagement. I also serve as the Oncology Consumer Health Information Professional in the Samuel J. Wood Library. In these roles, I provide evidence-based decision navigation interventions, information counseling services, and health literacy awareness education to promote informed decision-making among the oncology community. We have an exciting lineup of 3 speakers for you tonight. First will be Dr. Guy Maytal from Weill Cornell, speaking on the psychosocial aspects of genetic testing. Next is Dr. Nicola Rubin from the New York Genome Center, speaking on genomic testing and family history of disease. And last but certainly not least, we have Ms. Nyla Charles from the Meyer Cancer Center Patient and Family Advisory Council, offering a patient's perspective with genetic testing. To round out the evening, there will be a panel discussion and question and answer session consisting of all 3 speakers and facilitated by Dr. Andrea Betesh and Michelle Permiano of Weill Cornell's Clinical Cancer Genetics team. If you have any questions throughout the program or during the panel discussion, please use the Q&A feature at the bottom of the Zoom window. Tonight's session will be recorded and made available online for later viewing. In alignment with the Dan McMenamin Endowment mission, tonight's event strives to foster cooperation among patients, physicians, and researchers by educating patients on the complexities of collaborative care and offering resources to guide the patient in making informed decisions and navigating the health system. We hope that patients and caregivers of those diagnosed with or have the potential for diagnosis of rare cancers, as well as physicians and researchers in the rare disease space, will find tonight's program enhances their knowledge and improves patient-centered care delivery. Without further delay, I would like to introduce our first speaker, Dr. Guy Maytal. He is currently an assistant professor of psychiatry and serves as the chief of integrated care and psychiatric oncology at Weill Cornell Medicine, New York Presbyterian Hospital. Dr. Maytal was educated at Harvard College and Johns Hopkins University School of Medicine. He completed residency in adult psychiatry at Massachusetts General Hospital and fellowship in psycho-oncology and psychosomatic medicine at the Dana-Farber Cancer Institute, both in Boston, Massachusetts. He later served on the faculty of Mass General Hospital and Harvard Medical School for 11 years, where he developed and expanded multiple clinical programs in the areas of general psychiatry, psychosomatic medicine, and palliative medicine. Dr. Maytal joined the faculty of Weill Cornell in 2018, where he continues to develop innovative clinical programs, teach students and peers, and provide clinical consultation and care. His work focuses on developing a division of psycho-oncology, as well as developing integrated models of mental health care delivery. Dr. Maytal has written widely in his areas of clinical expertise. He has authored and co-authored peer-reviewed papers, review articles, and book chapters in various areas of psychosomatic medicine. Furthermore, Dr. Maytal is a dedicated and passionate educator who is actively involved in the teaching and supervision of medical students, residents, fellows, and staff physicians. Please join me in welcoming Dr. Guy Maytal.
My name is Guy Maytal, and the title of my talk is Dancing with Uncertainty, Important Decisions and Limited Information. So I'm going to be talking us through how the nature of uncertainty, especially when it comes to making important decisions, as well as what are the causes of uncertainty, what do we mean by uncertainty, and then how do we deal with it in a way that's useful in approximately 15 minutes or less. So let's just jump right in. So I have no financial disclosures. And I want to talk about, when I say uncertainty is, right? What is uncertainty? Or what is the valence, the emotional valence, or the initial impression that we have around uncertainty? And for most of us, it's like uncertainty is unwelcome. It's bad. We don't like it. And certainty is good. We want things to be certain, calm, easy, know what to do at every moment in time. However, when you really look at it and examine it, to be human is to be uncertain. As John F. Kennedy said, there's nothing more certain and unchanging than uncertainty and change. Or to quote the philosopher Francois Voltaire, uncertainty is an uncomfortable position, but certainty is an absurd one. So here's an example that might sound familiar to some of you. Joanne is a 37-year-old married woman in the process of undergoing in vitro fertilization, or IVF. Her father, unfortunately, is diagnosed with pancreatic cancer and has genetic testing that reveals BRCA2 as one of the genes associated with his cancer, which, as some of you know, is associated with both breast cancer and ovarian cancer, as well as pancreatic cancer. And the question Joanne has is, should I be tested for BRCA2? But not only that, should I start to undergo screening for pancreatic cancer, whether or not I undergo testing? And most immediately, should I undergo pre-implantation genetic testing for the embryos as part of my IVF? These are not straightforward or simple questions, and I offer no answers other than to point to the uncertainty that they evoke. So let's talk about uncertainty. And there are a couple of things we have to get clear about, because there are several things that get kind of mushed together. Uncertainty versus risk versus anxiety. And all three kind of come together around decisions like the one Joanne has to make, but they're actually very distinct. So uncertainty is the quality or state of not having certain knowledge. It is a state. It is a state of being. Risk simply refers to the possibility or the odds of something bad happening. It is a fact about probability or odds. And anxiety is a feeling of worry, nervousness, unease, usually about some upcoming or imminent event or something with an uncertain outcome. But again, they are distinct. Uncertainty is a state of being. Risk is simply a fact. What are the risks? What are the odds? Whatever that means, we'll get to that. And anxiety is a feeling or a feeling state. So let's talk about uncertainty and the human condition, right? Uncertainty is a function of free will. All of us really want to be free to make our own choices, to make our own decisions in life. No one's telling us what to do most of the time. We run our own lives, certainly as adults, despite sometimes our complaints to the contrary. See, the funny part is, if certainty at every moment, in every situation, either means no freedom or omnipotence. And in a strange way, that means no freedom. There's no freedom to choose, no freedom to agree or disagree, and no freedom to make mistakes. So as part of the human condition, when I said to be human is to be uncertain, the freedom to make your own choices requires uncertainty. There's a quote from a wonderful novel called The Magician King, where one character says, "I don't think the gods can change their minds." When you get to that level of power and knowledge and perfection, the question of what you should do next gets increasingly obvious. Everything is rule governed. All you can ever do in any given situation is the most gloriously perfect thing, and there's only one of them. Finally, there aren't any choices left to make at all. You're saying the gods don't have free will? The power to make mistakes, Penny said, only we have that, mortals. So uncertainty is innate to the human condition, to being human. It's with us all the time, though we don't experience it most of the time. And that's interesting and noteworthy. How come, given that everything mostly is uncertain, we don't experience all that anxiety that goes along with it in certain cases? So when confronted with a decision that we don't know what to do, the question is, how do I decide? How do I cope with the anxiety that goes along with it? Well, we've got to get to the cause of uncertainty. See, here's one possible approach. It's actually a pretty good one. You can see it says on the top left, embrace uncertainty. And then ask the question, what is to be done next? And then do that thing, and then repeat. Now, easier said than done, especially this. What is it to embrace uncertainty? It's kind of used in various settings, like self-help settings, or you'll find it in an all too facile memes online. But let's talk about what that actually takes and what it actually looks like in reality. So the first thing to get is that uncertainty is a function of the brain. It's only because we have a brain that can assess, that engages with the world in a particular way, that we even have a notion of uncertainty. You see, there's no uncertainty for the Earth regarding earthquakes. Earthquakes happen. The Earth doesn't say, I'm worried that there is a 50% chance of an earthquake today. Human beings say that. It's a function of the brain. You know, the brain has one job. The brain's purpose is to keep us alive. Right? You've got John Travolta staying alive and Gloria Gaynor's I Will Survive. The brain has one job. It's not to think, it's not to be innovative, it's not to create, it is to keep you alive. And it's pretty impressive that it's worked 100% of the time so far. So far, no one here has stepped in front of oncoming traffic. No one has fallen into an empty elevator shaft, right? No one's eaten poison inadvertently. Like, the brain works perfectly. Now, that's true for every mammal, and most animals that have a brain. That's the job. And for us human beings, the brain's job is also to preserve and keep alive our sense of self, who we think we are. So how does the brain work to mitigate uncertainty? How does the brain ensure survival? The brain is a prediction machine. That's all it does. It predicts stuff. Information comes in through your senses. Your brain compares it to stuff it already has stored from your past experiences, and then says, this new thing is close enough to this old thing. Aha, it must be just like this old thing. I'll do the exact same thing that worked last time to survive, so I will now take that action. So in a very real sense, there's no new information coming in because everything is seen through the filter of what you already know. Your brain predicts everything. You wake up in the morning, and when you go to the mirror, you're not surprised by what you see. Well, maybe you are, but you know what I mean. You're not surprised by you. You go, there I am. You don't go, my God, who's this in the mirror right now? It predicts everything. There is some possibility that you wake up and you find something new, but mostly not. Because mostly your brain knows what to expect. You know who's at work. You know how to get to work, even if that's going to another room these days. You know the people in your life. You know what they're going to say. You know those Thanksgiving conversations are always the same every year. There's nothing. It predicts nothing. It predicts everything. And so mostly we're surprised by nothing. Notice how little we're surprised. You don't walk out, oh my God, look at that tree. That tree looks slightly different than yesterday. No, your brain doesn't care because it's not a survival question. The brain works very well. In fact, it works better than any supercomputer yet developed, for example, to predict people's faces. All you need is a tiny bit of information that comes in, and your brain does the rest. It fills in the gaps. It works very, very well, except when it doesn't. There is this example, something called Schrodinger's cat. It's a thought experiment came up by the physicist Schrodinger in the mid-1900s, where he said, I put a cat in a box with a device that over the next hour has a 50-50 chance of killing the cat. So is the cat alive or dead? And the only way you know for certain is you open the box. But until you open the box, you actually don't know. And in fact, Schrodinger concluded that the cat is both alive and dead at the same time. The only time that probability collapses to one or the other is when you open the box. It seems absurd, and initially he intended it to be absurd, but the Schrodinger's thought experiment underpins a lot of probabilistic thinking, and the brain can't handle that. Another thing the brain can't handle is when it's biased towards seeing threats, even when there are none. You see the top left, what do you see? Looks like that, yes? Well, if you look again, you see a cute little kitty cat with a shadow. Because again, your brain is primed for survival. So it would rather err on the side of caution. So your brain isn't good at discerning between threat and not threat when there's ambiguity, and it's terrible at probability. See, your brain's not designed to think explicitly in terms of risk or probability. Your brain wants to know, is it going to kill me or not? Is there a threat or not? Am I going to be embarrassed or not? So it turns everything into binary decisions. And by the way, it's almost always going to err on the side of avoiding risk, almost always. However, genetic testing, especially for rare conditions, and the consequences of that testing are not binary, they're probabilistic. There's a 30% chance this will show up at some point in your child's life. That's very different than your child is going to live or die. Completely different worlds. Your brain can be trained to think in those terms. Most of our brains, we do not train them to think that way. And when we encounter something that we're not trained to manage, like probability, it brings uncertainty, which is always around us, right from the background into the foreground. In other words, your brain stops being able to predict. And the moment you can't predict, I don't know what to do with this. You're telling me there's a 70% penetrance lifetime in my child if they have 2 alleles. But what if they have only 1 allele? Or what happens, which is more effective, getting ongoing screening? Or do I need to have bilateral mastectomies? Do I need to have my pancreas scanned every 3 months, 6 months, 9 months, 1 year? What are the odds? The brain isn't good at that. So that's why all of a sudden anxiety arises. There's a sense of threat when the brain stops being able to predict. And it's fight, flight, or freeze, right? Like something's coming to eat us. But that's usually insufficient in the face of chronic stress. What happens is, unfortunately, you stop thinking. You just got to survive. Your brain's focused on how do I avoid being eaten or killed? How do I get through this? We disconnect. Hard to think in those moments. So now here we are to embracing uncertainty. What I mean by that is really granting yourself the space to be, however you are. For some people, it's this. Like, I've got no idea what's going to happen, and I love it. For most people, it's more like this little feline. I'm going to take a little step out into the world, even though it's uncertain. I don't know what to do. My brain can't manage this, and I'm scared. But wherever you are on the spectrum, that's okay. The point is, embracing uncertainty isn't changing your anxiety. It's giving your anxiety space to be, just as it is. So what do? You practice. See, as Bobby Knight, one of the most successful college basketball coaches, said, everybody wants to be on a championship team. No one wants to come to practice. What do you practice? You practice whatever there is to do to manage your anxiety. You've been anxious before, you've gotten through it. Do what's worked in the past. Talk to people, get information, get reassurance, get coaching or mentorship, exercise, avoid the thing for a while, practice mindfulness. The point is to keep practicing. See, the challenge with probabilistic information and choices, like around genetic testing, is that there's no right or wrong answer. There's no, if I do this, I'll be eaten or I'll die, and if I do this, I won't. That's why the brain gets so short-circuited. See, ultimately what you do is you make a choice and you live with the repercussions. But that's what you do every day. You make choices around food, you make choices around clothes, you make choices around jobs, you make choices around where you live and who you'll partner with. And you say to me, but this isn't life or death. And you're right, but neither is this. This is probability. You see, your brain wants to turn it into life or death. Do I buy a safer car or a sexier car that may not be as safe? That could be a life or death decision. We don't frame it that way. And the other two things is, one is take care of yourself, be human. In the face of anxiety, humans need physical needs met. Humans need to enjoy things that they enjoy. Humans can be however they feel. They feel sad or happy, angry, tired. Just allow space for all of it. And we're social creatures, especially nowadays when our social interactions are much more limited, connecting with others. And there are multiple categories of coping with anxiety that I've already mentioned. Moving, caring for your body, mindfulness, connection to nature, animals, or people, or just living with a purpose. And finally, here are additional practices. Practice letting go of what you can't control. Again, it's hard, but that's why you have to practice it. Allow yourself to feel what you're feeling. Stop trying to make anxiety stop, because you can't anyway. And then practice focusing on what you can control. And ultimately, practice being brave. In other words, feeling the fear and taking an action. Any old action. As Amelia Earhart said, courage is the price that life exacts for granting peace. So, in summary, uncertainty exists by virtue of our brain. The brain predicts to survive and gets anxious when it can't. And probability is essential to genetic testing, and most brains are not trained to think in those terms, which leads to anxiety. And we also reviewed several practices to manage anxiety. And the key is that word practice. And expect failure. When you start something new as a practice, you'll fail at it. But keep practicing, as opposed to expecting perfection and stopping the first time you fail. Because ultimately, there's this lovely quote, just when the caterpillar thought that the world was over, it became a butterfly. So thank you for your time and your attention, and we'll have questions as part of the panel discussion. Thank you very much.
Thank you for that inspiring and insightful talk, Dr. Maytal. Our next speaker this evening is Dr. Nicola Rubin. Dr. Robine joined the New York Genome Center in April 2012. He graduated from an engineering school in Paris with a master's in bioinformatics from Brca, Sweden, and obtained his PhD at the Institut Curie in Paris for his work on the analyses of meiotic double-strand breaks in yeast. He did a postdoc at the Sloan Kettering Institute in New York in the lab of Dr. Erica Lai, working on the annotations of Drosophila small RNA. At the New York Genome Center, he initially developed the RNA sequencing analysis pipeline. As director of computational biology, he currently manages a group of scientists and analysts with projects applied to cancer genomics, functional genomics, and neurobiology. Welcome, Dr. Rubin.
Hi, my name is Nicola Rubin, and I'm director in the computational biology department of the New York Genome Center. Thanks for inviting me and giving me the opportunity to talk about cancer genomics today. Let me introduce myself and give you some definitions. I worked in what we call computational biology or bioinformatics. I was originally trained as a computer science guy back in France around 2000. In 2001 was published The Sequence of the Human Genome, the long stretch of 3 billion letters that we all share. Most of these letters are identical to all humans, but some are variable. In fact, enough of them are variable to make all our genomes slightly different from each other, making each of us unique, with the exception of identical twins. When the reference sequence was published, a lot of people talked about the need for computer guys like myself to get into biology and apply programming and computer tools to analyze biological data and contribute to our collective understanding of biology. That sounded great to me, and I decided to specialize in the field. I obtained a Master of Science in Bioinformatics, then a PhD in Paris, and arrived in New York in August 2008. I worked at the Sloan Kettering Institute for 3 years before joining a new center dedicated to the generation and analysis of genomics data in New York and called the New York Genome Center. The New York Genome Center was created by 12 academic institutions and hospitals in the New York region to be a place for large-scale sequencing and bioinformatics analysis. We have about 200 employees, 8 faculty members with lab co-affiliated with another university in the city, Columbia, NYU, and Cornell currently, a production lab with state-of-the-art sequencers, and a New York State certified clinical lab that has the ability to obtain samples directly from doctors and return genetic information that can be included in clinical records. Scientists in New York and elsewhere collaborate with us to sequence their sample and contribute to the analysis and interpretation of the results. I won't go into details of the sequencing technology for this talk, but here is what you need to know. Your genome is present in 2 copies, one coming from your mother, one from your father, in every cell of your body. You have hundreds of billions of cells in your body. They have different shape, different function. Some are in a fixed state, some are differentiating into specialized function, some are dividing, and some are staying the same for most of your life. In each cell, there is a specialized compartment called the nucleus, and in the nucleus are chromosome, which is form of DNA, short for deoxyribonucleic acid. It's a very long molecule formed of 4 different building bricks called the nucleotides, and that we represent with the letter A, C, G, and T. Sequencing a genome means taking cells of your body, extracting the DNA, and reading these long molecules. We can do it from any kind of cells, but the easiest to collect are saliva cells, skin cells, or more commonly blood cells, which have less contamination from other organisms, like the bacteria that live in your mouth or in your skin. We read the DNA molecule with an instrument called a sequencer, and the one we mostly use are manufactured by a company called Illumina, based in San Diego. It splits the molecule into small fragments of a few hundreds of letters. And for a human genome, we generate many billions of those small fragments that we call reads. The first step of the bioinformatics process is to find where these fragments are located in the reference sequence and to identify the small difference between a sample and the reference sequence. Every 1,000 nucleotides or so on average, we find one letter that differ from the reference and we mark that difference. Then we collect all these differences that we call variants, and we identify a sample by the places where it differs from the reference. One reason to do this is to identify differences associated with a particular disease. We can collect samples from a population of affected individuals for a disease and compare with a matching population that is not affected by the disease we're interested in. If the disease has a genetic basis, we can identify the variants that are associated to the disease. They can either cause the disease directly or predispose the individuals to the development of that disease. This plays out at the molecular level within the cells of our body. Sometimes one single copy of a deleterious variant causes the disease. Sometimes both copies are needed to be affected. All diseases are the results of the interaction between the genome and the environment. What we eat, our exposures to chemical, to radiation, our physical activity, and so on. That explains in part why some people with a deleterious variant will develop a disease, and some with the same variant will not. The genetic information is not deterministic, but it influences the risk of developing disease. And by the way, what I describe here for disease is true for a lot of things, including our size, the shape of our face, the color of the eyes, or the hair, etc. We all know that these traits are in part heritable, and that people in the family tend to share some of these characteristics. So when the doctor is asking you about your family history of cancer, what they're indirectly asking is the possibility of carrying a genetic variant predisposing to the development of cancer. And I put here on the right side of this slide what we call the GWAS catalog. So all the different places on the 22 chromosome of the human genome that are known to be associated with a disease or a trait. And so it's an ongoing effort. That's something that a lot of people are doing in the field that we call genetics. We know about 2,000 association between genetic variant and the risk of cancer. The most well-known and well-established predisposition gene is called Brca, which stands for breast cancer gene, because some variants of the gene were identified in families with a large number of women developing breast cancer. However, we now know that these variants also predispose to prostate cancer, ovarian cancer, and other types of cancer. We also know that the role of these genes is not to cause or to prevent cancer. The protein produced by the Brca gene are playing a role in DNA repair mechanism. I mentioned the hundreds of billions of cells in our body and the fact that most of them divides. The way they divide and the rate at which they divide is very well regulated, which explains why our body remains more or less the same shape and size during our life. Some cells die, some new cells are created, but more or less our total number of cells remain the same during our adult life. During cell division, the genetic material is duplicated and then divided into two daughter cells. During this process, and other process within the life of the cell, some errors can occur and some mutations be introduced. However, evolution developed sophisticated processes to correct these errors. Some sort of a quality control mechanism, very powerful, but not entirely 100% perfect. Occasionally, some errors go through. One element of this quality control is just a little bit less efficient, a little bit more errors go through, and that increases the likelihood that this error will affect a critical element of the genome. And as error accumulates, the possibility that the quality control processes of the cells get damaged increase, which can lead to proliferation of the cells. They divide more and more, they accelerate, they accumulate more and more mutation, and they escape the quality control and the regulation system that normally function in the cell. So by comparing genomes of people affected with a particular form of cancer with matching genomes that do not have the cancer, we can identify some overrepresented variants that we can try to understand what role they have in increasing the likelihood of cancer for the people with these variants. Some variants increase the likelihood of cancer a lot, up to 80% of developing cancer during the lifetime of individual, for instance, in what we call Lynch syndrome. And some variants have a much smaller effect. In these cases, few individuals with the variant actually develop the cancer. We can have a predisposition variant and not develop cancer. And conversely, we can develop cancer and not have in our genome any of the known variant of predisposition. There are multiple ways in which the quality control processes of the cells can be deregulated, and the role of the genetics can be very small in some cases. Because we inherit our genome from our parents, we receive half of our mother's variants and half of our father's variants. If one of our dad's variants predisposed for cancer, we have a 50% chance of having it as well. But having one parent or a sibling with cancer does not necessarily imply that we will develop cancer as well. We need the work of doctors to identify the frequency of cancer in the family, which type of cancer, and determine the chance of having a familial form of that cancer, or what we call a sporadic form. One other application of the sequencing technology is the sequencing of the tumor. As I described, tumor developed by proliferation and accumulation of mutation within the cell. As a result, a full-grown tumor contains a large number of cells, usually with a large number of mutations in their genome. We call these mutations somatic because they were formed during the lifetime of the cells and were not inherited from the parents. By sequencing the normal genome in non-tumor cells and the tumor genome in tumor cells, we can identify all the somatic mutation in the tumor. We can classify certain tumor according to the type of mutation they have. For example, we know of at least 3 subtypes of breast cancer, and they are largely different disease, different treatment, different outcome, different frequency of occurrence, etc. We can also identify somatic variants that can be targeted with certain drugs. This targeted therapy will kill the cell where the somatic variant is present and leave unaffected all the other cells. Knowing all of the somatic variants in a tumor is also important to study the tumor, to know its evolution, what variants are recurrent in that cancer type or rare, to understand which of the quality control processes I talked about earlier is deficient and need to be fixed. Even if the tumor is in a different organ, it can share a lot with another tumor type for which much more is known, and maybe even for which there are available treatment options. And finally, it can be important to estimate the overall number of mutations in a tumor because we know that on average, immunotherapies work more efficiently in highly mutated tumors than in tumor with fewer mutations. In the context of rare cancer, genomics can unravel a large amount of information about the tumor. One particularly striking example is a rare liver cancer affecting adolescent and young adults and called fibrolemular hepatocellular carcinoma. There are about 400 cases every year, and little was known about this disease until we sequenced the tumor of 10 patients, which led to the discovery of a single mutation present in all of the cases. This is now the defining hallmark of that disease, has opened up the possibility of several clinical trials, and has completely changed the research activity around this particular cancer type. Cancer genomics provides scientists and clinicians with a very accurate description of the tumor, much more detailed than what can be obtained with other methods, and than what is known just by knowing the tumor type or the organ. Each tumor is unique in the sense that the assortment of somatic mutation is unique, and the interplay between the somatic variant and the genome of the patient is also unique. Clinicians can take advantage of this knowledge to tailor individualized treatment in what we frequently call precision medicine or personalized medicine. I would like to close this presentation by talking briefly about two projects I am leading at the New York Genome Center. One is called Vrcc, for Very Rare Cancer Consortium, where we propose to identify cancers so rare that even the large cancer centers in New York struggle to study and to assemble cohort of patients. By pulling together these resources and the patients from all major hospitals in the region, we can create this cohort, enroll enough patients, and sequence the tumor of this understudied cancer type. We then assemble teams of experts across the city and discuss the results in a tumor board setting. We started these meetings last year and have had tremendous response from clinicians and from patient advocates. We're looking for funding and are looking forward to start enrolling patients in this research study. Another project is called Polyethnic 1000 and is based on the realization that most cancer genomics, and indeed most knowledge about cancer, has been derived from tumors of white patients. However, we know that some cancer behave differently in different ethnic populations. They have a different frequency, different outcome, different response to treatment. Some of these can be due to differences in diet, lifestyle, socioeconomical conditions, but it is also known that some of it can be associated with genetic differences between populations of different ancestries. We want to take the opportunity of the great ethnic diversity in New York City to study the molecular basis of these cancer disparities. And we announced in September 2020 the start of 6 projects focused on breast, lung cancer, pancreatic, bladder, colon, and endometrial cancers. This is the end of my presentation, and I look forward for the discussion and for any question you can have about cancer genomics in general. Thank you very much.
Thank you, Dr. Rubin, for that informative talk on genomic testing and family history of disease. Our final speaker of the evening is Ms. Nyla Charles. Ms. Charles was a former global management consultant before finding her true passion as a writer. Her debut book, Mama's Afro is a Soldier Too, A Mom's Cancer Diagnosis Explained, is a children's book inspired by her own cancer diagnosis. Ms. Charles is a dear friend and volunteer to Weill Cornell and holds several volunteer roles. She is a co-chair of the Patient and Family Advisory Council of the Meyer Cancer Center, the patient advocate on the Breast Management Leadership Committee of the National Accreditation Program for Breast Cancers, and is a patient advocate for Cornell's research projects within the cancer epidemiology space. Ms. Charles is also the vice president of the New York chapter of Sisters Network, Inc., the biggest national organization dedicated to spreading awareness and education of breast cancer within the African American population. Her biggest role, however, is as a doting mom of her 2 lovely daughters. Please welcome Nyla Charles.
My topic is, it may not be rare, but it is no less significant. And before I dive into the real meat of what I wanted to talk about today, first I wanted to talk to you and give you a little bit of information about my family. So meet my family. My family consists of my father, my mother, their daughter, then me, another daughter, a 4th daughter, and a 5th daughter. My parents have 5 daughters. Poor dad, right? Then the other thing I wanted to go through is to really let you know a little bit about our health history, and in particular, our cancer history. So our cancer history is such that my father passed away in 2014 from prostate cancer. My mom, who's with us thankfully, was diagnosed 15 years ago with breast cancer, and then 3 years ago with ovarian cancer. This is me. and 3 years ago, I was diagnosed with breast cancer. When we were diagnosed, my mom and I, we decided to do some genetic testing. So this is our history. We did a number of genetic tests, a very comprehensive panel of genetic testing to see our predisposition for different types of cancer. And just because she had breast and ovarian cancer, there was a higher suspicion that she was BRCA positive. So she did the test, and she was found to be BRCA2 positive. I also did the test, and I was found to be BRCA2 positive. So I spoke to my sisters, and I asked them if they'd be willing to do the BRCA testing, just so that they can find out if they also had a predisposition for any of these cancers due to the mutation in the BRCA gene or any other gene. A very good doctor at New York Presbyterian was doing a research study, and she offered to actually have my whole entire family tested, which was great because we were based in England as well as in the Caribbean. So I spoke with my first sister, and she agreed, yes, she would do the BRCA testing. The third sister said yes. The fourth sister said yes, but the fifth sister said no. And it was a little bit of a surprise. I mean, in her mind, she just didn't want to know. And she said she doesn't think anything is wrong, and she feels fine, and she's not going to do the testing. But after some convincing, we convinced her to do the testing. So the results were like this. So in terms of my first sister, she tested negative. Obviously, I tested positive, as I mentioned before. My third sister, she tested positive for the gene. My all-partying sister tested negative. And my fifth sister tested positive for the BRCA gene. So what does that mean? So now what? Now what do we do with this information? BRCA, everybody has a BRCA gene. And obviously there are more doctors on the panel to explain the medical side of things, but everybody has a BRCA gene, but the mutation on it in those genes cause an elevated risk of cancer. So with the BRCA2 gene, we know that it causes an elevated risk of breast cancer, as well as ovarian cancer, pancreatic cancer, melanoma, and the geneticist had also spoken to me about the risk of thyroid cancer. So what do I do with this knowledge? What does anyone do with this knowledge when they have a BRCA2 mutation? Well, my sisters and I, we know that we have a higher risk of breast cancer. So what's advisable is to get mammograms and ultrasounds annually, and also get breast MRIs annually. And we can spread that out at 6 months at a time. We can spread it out across 6 months. And in that way, we can have 2 sets of imaging done in 1 year. We can also decide to have a prophylactic mastectomy. And that's what I did. Even though my breast cancer was just in one breast, I decided to do a double mastectomy just because of my increased risk. Because of our increased risk of ovarian cancer, we can do more screening, like ultrasounds, we can do blood work, we can even do a prophylactic oophorectomy, which is the removal of our ovaries and our tubes. Just last week, my sister in England actually had an ultrasound done just to actually do as part of her screening actually for ovarian cancer. For pancreatic cancer, what I do is I do an endoscopic ultrasound, and then I alternate it the following year with an MRI. And it's what we need to do to see if there's anything that could be irregular or anything that could be caught early. I know that there's no ideal screening for pancreatic cancer or ovarian cancer, but the screenings that I've indicated are about the best that is available right now. The other type of screening that I do is for melanomas. I get an annual skin check with my dermatologist. And it's a full body check to see if there's any lesions or irregularities or moles or anything suspicious. And I also get thyroid screening. So actually last week I got a thyroid ultrasound, and as well as I do some blood work as well. And so all this is to say that, you know, breast cancer is not a rare disease, but it's still very significant because the breast cancer that my family, my mother and myself were diagnosed with, it was born from having a mutation in the Brca2 gene. And for us to actually get the information about our cancer history and the genetic history, it just makes us more on top of our health. We're able to advocate for ourselves. We're able to monitor it closely, our health closely with the doctors. And we're just giving ourselves the best chance of detecting anything early on. And so that to give ourselves the best chance of getting something treated early on, if that happens. It's not fun. It's not anything anybody wants. I feel I know New York Presbyterian and Cornell like the back of my hands. I can close my eyes and walk through any building. I feel any building that I don't know, I'm surprised because I go to everyone. I go to the breast center. I go to the gynecology center. I go to the gastro. I go to endocrinology. I go to dermatology. Plus, you know, you have your normal PCP and your eye exams, and you know, but I just feel better that I'm trying to give myself the best possibility to survive. I have two little girls, two daughters, and they're young. They're 8 and 6, and I want to make sure that I'm here for them as long as God lets me. And the other thing, I know because of my history, I want them to be, I'll be more mindful that when they get older, when they get to be adults, that they can also have the same type of testing to see what their risk is for any cancers, so that, you know, we just survive as much and as long as we can and live. Thanks very much. You know, I mentioned Angelina Jolie and how she was the first person that I heard about who did a double mastectomy because of this Brca2 gene mutation. And it's one of my biggest regrets about not doing anything about it. I remember that time I thought, oh, let me go and check it out. And I had it on my list, to-dos list. But then life got in the way, everything got in the way, and then I just didn't actually, I didn't explore it. And it turned out that obviously that I got breast cancer. But had I known before, then I would not have had to go through all the treatment or the support or the radiation, surgeries and everything that I did. And I would not have had to find a difficult a way to actually speak to my young kids who were just 5 and 3 at the time. I would not have had to have a difficult conversation with them. And I would not have even had to write this book that I wrote. I wrote a children's book called Mama's After as a Soldier Too, which you should see on the screen. And that book was one that I created just because I needed to find a way to tell my young kids about their mom having cancer. And I'm very proud of the book. It's a much needed resource, but I would not have had to write it if I didn't actually have breast cancer. And if I had to go back in time, I would prioritize self-care and looking and being on top of my health, even though I was pretty healthy, just because it's a difficult thing to go through. It's a difficult thing for any cancer to go through. I got through it, and I'll continue to get through it, continue to go to my doctor's appointments. But if I leave you with one thing today, it's to say, just be on top of your health. It's so worth it, because nothing else matters after your health.
Thank you, Nyla, for sharing tonight. Your story is powerful and invaluable to the conversation. Now we're going to move into the panel discussion portion of the program. I want to welcome back all of our speakers and our two facilitators for tonight's discussion, Dr. Andrea Betesh and Michelle Primiano. Dr. Betesh is an assistant professor of medicine in the Division of Gastroenterology and Hepatology at Weill Cornell Medicine. She practices general gastroenterology with a focus on the care of patients at high risk for gastrointestinal malignancies and hereditary cancer syndromes. Other areas of focus include quality improvement in colorectal cancer screening and endoscopy education for GI fellows. She received her undergraduate Bachelor of Science in Neurobiology from the University of Texas in Austin in 2008 and graduated from the University of Miami Miller School of Medicine in Miami, Florida in 2012. In 2015, she completed her internal medicine residency at Baylor University Medical Center in Dallas, Texas, where she also served as chief resident. She completed her gastroenterology fellowship training at New York University Langone Medical Center in 2018. In 2020, she completed the City of Hope intensive course on genetic cancer risk assessment. Michelle Permiano is a genetic counselor for the Clinical Cancer Genetics Program at Weill Cornell Medicine. Ms. Permiano is certified by the American Board of Genetic Counseling and holds an advanced certification in pharmacogenetics from Manchester University. Ms. Primiano served as a clinical genetic counselor in pediatric genetics and coordinator for several genodermatosis clinics at Columbia University Medical Center prior to joining Weill Cornell. She also serves as a clinical director for the Long Island University Genetic Counseling Graduate Program. Ms. Primiano has dedicated her career to advocating for and providing access to clinical genetic services for all patients and educating genetic counseling students and healthcare professionals. Dr. Betesh, Michelle, the floor is yours.
Hi, thank you so much for having us, and thank you to all of our speakers. We have a few questions for our panelists, and I'll start with you, Ms. Charles. One of the things you mentioned is that as your daughters grow up, you're gonna tell them about your history in more detail so that they themselves can get tested one day. And one of the things that comes up a lot is how to talk to young adult children about genetic testing and genetic mutations in the family. And I'm curious if you've given thought about how you will have that conversation with your daughters as they get closer to the age in which they may consider testing.
Yeah, sure. I mean, right now, since my kids were 5 and 3, I didn't have a resource to talk to them about the breast cancer. But so I spoke with them very frankly and in an age-appropriate manner that something was growing inside of me. And then I needed to get it tested. So, and then I also had to, because I've written a book, like turned lemons into lemonade. My kids have, they've been my beta readers, so we talk, they hear me talking about cancer a lot. They talk about cancer a lot. They're very aware of I guess, in terms of in their age appropriate way, at their level, what it is. They know it's not something great. So for me, it will be easy for me because I've had that conversation with them. I've written the book, they've read it, they've reviewed it, they've given feedback on it. So for me to explain to them that there's a possibility that they might have a gene that will predispose them to having it, it's just another step in the story, another chapter in the story that they already know about. So for me, I don't think it's going to be difficult or complex for me. I've tried to be very, very open with my kids about cancer.
And Dr. Maytal, in your experience for patients who are getting this information and maybe have not been open with their children, how would you sort of counsel people on how to breach this conversation with young adult children whose lives and health may be impacted by this information?
I would actually defer to Ms. Charles's experience, but all I can offer you is it completely depends on the age of the child, the degree of maturity. It depends on how directly their lives are going to be impacted by it, right? So a diagnosis of a Huntington's disorder gene is very different than something with much lower penetrance. It's part of it, really is a dance in the relationship, which is given by your knowledge of the child and the type of relationship that you have with them and how old they are. So I think it's a little bit too general a question to give any specific responses to.
So we do actually have a question from one of our participants. It's, what are the steps to getting genetic testing? And I'm not sure if anybody wants to share their thoughts on how people should go about this.
So I think as a provider who performs genetic testing, I think I can comment on that. One of the best ways to get genetic testing is through your physician or genetic counselor at the facility where you get your medical care. I think that a lot of patients may pursue genetic testing in other ways as well, because there is a lot of direct to consumer testing companies. However, I think that if patients want to really understand their genetic cancer risk, getting that testing in the context of a genetics provider relationship is really important, because you can order, you know, a saliva swab through direct to consumer companies, and I won't name any of them specifically, but what you're really lacking if you do that is the counseling piece of it that occurs both before and after testing, because this testing really has a lot of implications for the individual getting the testing and also for the family members. And so I think it's important that people speak to their doctor rather than trying to navigate this on their own. I think we also just got another question in the chat box about whether primary care physicians are generally sending genetic testing or should patients without cancer but a strong family history, should they self-refer? And I think if you're concerned, the first place to start is to start with your primary care doctor. And if your primary care doctor is concerned by your family history, they can certainly refer you. But if you yourself know that you have a strong family history, you can also seek out a trained genetics provider to be evaluated and find out, you know, should you get testing, what would the potential implications for testing be, and how would you handle the information that you received from the testing.
I think another important point to add to that, Dr. Betesh, is people may have no personal history of cancer, but a strong family history. And even if they pursue genetic testing and the results come back normal, they may still be eligible for what we call high-risk surveillance. And that's an important thing to mention, because just because there's no identifiable genetic reason for the cancer in your family does not mean that you wouldn't qualify for more increased screening. And that's an important conversation to have with your healthcare providers.
Yeah, I think that's a really great point, because a lot of patients don't know that some of the criteria for enhanced screening is really more about the family history and other risk scores that your provider can calculate for you. And it's not just about the actual genetic testing results. There's a lot of factors that go into an overall risk assessment. And I think that that's important for patients to be aware of, that it's not just the genetic testing, but it's the whole picture of the patient.
So, sorry, this is Nicola Rubin, and I am not a provider. I can just add a few more things on that. One of them is that from a technical point of view, the quality of the test you will get in the clinical setting is certainly higher than you would get in a direct to customer to consumer approach. It will look cheaper, but the quality will not be as high, and so you will get a much higher sequencing and genetic test overall if you do it in the clinical setting. The other aspect is that it could well be that the cause for the family history is genetic, but you will not test for it. When we do a genetic test, we look for certain places in the genome that we know and we identified and we can associate with a risk. And we will only look at those places. The cause for the high incidence of cancer in the family might be a genetic defect, but we just don't know about it, and we will not look for it. We will know nothing about those variants if we identify them, and we will not be able to provide counseling. So it's still a genetic cause, but we don't know about it, and we will not test for it, right? The genetic test that mostly we do is looking at the known and most common variant associated with those cancers.
And just to piggyback on that, Dr. Rubin, the other thing to think about is that as you know, the field of genetic testing is moving very quickly and the technology is rapidly improving. So some people who may have had some genetic testing many years ago and maybe tested negative, but if the family history is concerning, it may be worthwhile to talk about getting updated testing, which is something that is coming up as the testing becomes sort of more advanced and more cost efficient. Some patients who've had testing many years ago may consider doing that to sort of reevaluate their risk now.
So we do have some more questions coming in from the attendees. I think this one would be good for Dr. Robine. It seems that they're asking about updates on blood testing for the detection of cancer, so potentially circulating DNA, and being able to diagnose cancer at earlier stages. Do you have any comments on that?
I have, but then I have to disclose conflict of interest in the sense that I'm working on those tests, and that's one of the research activity that we're doing, and we have published a paper in June in collaboration with Weill Cornell Medicine, looking at the possibility to do blood tests. So I'll backtrack a little bit. Tumors, wherever they are located, are proliferating, generating a lot of cells, and some of those cells die and explode. And when they explode, they release their DNA, usually into the blood. And those fragments of DNA coming from the tumors are then circulating in the bloodstream. And when we sequence the blood sample, we might capture some of those fragments of DNA coming from the tumors. And we can find ways by sequencing, by doing genomics and bioinformatics and analysis to separate the normal DNA from the blood and the DNA coming from the tumor. And we think we can use this to track the evolution of the tumor. In particular, in our paper and in something that other people are doing after surgery, when we know there is a cancer, we know there is a tumor, we surgically remove the tumor, and we want to track if the tumor is going to recur, if there's going to be a resurgence of the cancer. And we can track this. Usually those tumors are regrowing and are really small, but we can track them in the blood, and that's what we try to demonstrate in our paper. And so that's one application. Obviously, the big application is the early diagnosis, right? It's the possibility of detecting cancer earlier than is currently possible just by doing a blood sample. And so there is a big company called Grail, and I'm not citing it to make any publicity, but just to say that's the holy grail of cancer genomics and cancer biology in general, is to have a test that would be relatively cheap, that would perfectly discriminate between people that don't have cancer and people that have a very early phase of cancer that will develop into cancer. And so there is a lot of discussions about the validity of this approach. Will that really happen? Will there be a lot of false positive? Will there be a lot of cases where we will tell people, well, there is something really concerning, and we want to monitor, we want to look very closely. How exactly is that going to be? What's going to be the performance of those tests? All of this are ongoing questions, but there are very clearly a lot of economical, a lot of money put into those tests, into those trials. And basically, ultimately, the goal is to have, we can imagine a world in which every 6 months, everybody get tested, and some of them are going to turn positive or will have a signal that will trigger some additional test or a closer regimen of testing, so that we can follow the evolution of this signal and identify and capture and cure the cancer before it develops into something really malignant. That's the whole idea behind the blood test. And I will not, hazard myself at giving you a timeline and a date for when those tests are going to be available, but that's what people are working on.
We have another question for Nila, and this kind of goes along with what happened in your family, where often we come up against family or friends who need a little bit of convincing to go forward with the testing. And what strategies did you use to help convince your sister to go forward and actually get the Vrca testing?
It was a collective effort, kind of like a sister intervention. Me from New York, my other sister in Trinidad, and the other one in England. All of us telling, the other two in England, all of us telling her that this is good to get done. It doesn't mean that she has to do anything about it. It means, we just need to know. And this is an opportunity that we're getting the whole family tested. And, if she doesn't want to be the only one who's not tested. I mean, we also told her, I told her, I still tell her to this day, because she's kind of not yet convinced about doing anything, because she thinks it's a risk, it's not a definite that something would happen. So, even up back then and today, I still tell her, I wish I had known before. If it's only one thing I could take back, from being an overwhelmed, overworked, busy mom, it's that I wish I had just gone to take that Brca test, because had I gone, then I would not have to do so many things. And I told her, you don't want to have cancer, even if you detect it early, it's not, you're forever patient, right? You are forever patient. You have to go and get your screenings, go and see a doctor. You know, I have to take medicines. There are side effects to those medicines. You know, there's a lot. So it's a combination, I guess, of telling her everybody's doing it, so please do it, or, you know, it doesn't hurt, you know, get it. So the whole family will be tested. And also kind of like a little bit of scare tactics, you know, like do it, because if you get cancer, you would regret it. So yeah, I think we did a few things. So now she did the test. So now it's the next stage is trying to convince her to do something more about it. So obviously I had cancer. My sister in England, she's doing. She's following up with the NHS, it's the National Health Service in the UK. You know, there are issues there because they're not doing any non-essential type surgeries for the next 18 months. But the thing is, she's on top of it, and she's having herself checked, like to do her ultrasounds and mammograms. So my last sister, she just needs a little bit of convincing now in terms of doing things that could help mitigate her risk of cancer, giving her Brca mutation status.
Yeah, and I think another thing that's important for people to understand about getting genetic testing is just because you get genetic testing, it's not a definite that you're gonna get a cancer. It just, it's a way to help you to better understand your risk so that you can be empowered as a patient. This is a great way for you to have more information so that you can do things to hopefully prevent cancer, or if not prevent cancer, then detect it very early, because the earlier these things are found, the easier they are to treat.
Right.
So I think it's not information to scare anyone, it's information to empower you to take control of your own health.
Yeah, exactly.
So there is another question, and I actually had the same question for you, Dr. Charles. And the question is, what's an early age to be tested for the BRCA? And I wonder, when you were talking about your daughters, what would be the age, the appropriate age for the testing?
What I was told, it's at I think 18 or 21. Definitely as an adult. They
As
would
an adult.
not. Yeah, they won't allow me to have them tested as kids because they don't. Just because they don't want parents having to make a decision for their children.
But so it seems to be a legal definition rather than a biological or medical decision or medical argument for that age. Am I correct?
Yes, exactly. So I mean, my little one, my 6-year-old, she had to. There was a couple of things that were going on with her at one point, and they wanted to test to see. She was seeing a geneticist, a pediatric geneticist, and so they wanted to see whether she had inherited 2 Brca genes, but they would not. And there's a test to test that, but they would not tell me. So she was negative for that, for the double, but they would not tell me if she was positive for 1, because they could not legally. They could not. So. Yeah, and it's fine. It's fine. I mean, I'm quite, you know, on top of my health. I'll be encouraging them to be on top of their health. You know, I'm the mother who doesn't give my kids too many sweets, you know, or I'm very health conscious with them. So I mean, it's just part of our trajectory in life to be healthy. So.
And generally, when we're doing genetic testing for genes that cause risk for adult onset cancers, the thought is that the individual, the child, should wait till they're an adult so that they could make the decision themselves of whether or not to be tested. And the only situation in which we advise people to have children undergo genetic testing is for conditions in which there may be a childhood onset cancer. And that's also obviously a difficult situation depending on the age of the child. If the child's very young and there's a risk for pediatric cancer at age 4 or 5, obviously the child's too young to really be involved in the decision. But if there's a risk for the cancer in the child's, 12, 13, 14, they do have some autonomy, should be involved in that discussion. But those situations get quite complicated, and often we have pediatric genetics involved in those cases, to help navigate those tricky issues. I think there's another question in the chat box about genetic testing to help guide cancer therapy. So so genetic testing to help determine what types of treatment someone with a cancer may get. And I think, Dr. Rubin, that may be something that you're equipped to answer.
Yeah, I just don't see the question. So I think, I'm sorry. It's okay.
It's from Mr. Nathan. So there's genetic testing available to see medications that are suitable.
Yeah, sure, certainly.
Cancer conditions.
So yes, they are, and that's actually one of the main reasons we do cancer testing in the clinic. And it's the cancer testing of the tumor. And so we talked a little bit about the difference between the germline testing, where we test for genetic predisposition, and the example is Brca. But the other component is the somatic testing. So testing the tumor and identifying in the tumor, are there certain genes that we know are frequently mutated in that tumor type, and for which we have a drug that that will target that particular mutation. And so we now have a lot more of such drugs that we can use in a particular cancer type. And so we identify the cancer, we sequence the gene of interest for that tumor, and when the variant is present, we give the drug. If the variant is not present, then we do not give the drug. The drug has a lot of side effects. It's not good to give the drug when we know it's not going to have any efficacy. When we know it might have an efficacy, then we give the drug. And so there is a lot more of such matching between a variant and a drug, and that's definitely one of the most exciting things that happened in cancer research in the last 20 years.
So I think another piece of the question is using one's genetics to determine medications that a patient would respond to better than another medication. For example, with psychiatric conditions, or potentially some other, regular medications like for blood pressure, or potentially for high cholesterol, or people that have other medical diagnoses. I think that question comes up a lot, is, are we right now tailoring medications to patients that, have these regular, more common medical conditions?
Yeah, I mean, I think the same approach that we take in cancer. So in cancer, we test the normal genome and the tumor genome. But for other disease, we have the same kind of approach. What we, the field is called precision medicine, or personalized medicine. And again, the ultimate goal is to have the perfect drug for the patient. And so instead of considering all of the patients with the same disease, take each patient individually and say, for your form of the disease, based on your genetic information, based on your lifestyle, based on everything we can know about you, here is the best drug, and we know it's much more likely to work. What we do at the moment is that we have someone coming, we have some symptoms, and we have a drug associated to it, and we know it works sometimes, but not always. And so if we can increase the precision and the matching, between all of that. So we need a lot more information, gather a lot more information about the patient, and have a lot more drugs available, and do a very good match between the two. And that's really where the field is going in many ways.
Mhm.
And
Oh, sorry. I didn't
no,
mean
no,
to
but
cut
there's another term that's important. Is called pharmacogenomics,
you.
where
Mhm.
we know of some genetic variant that affect the way we metabolize drugs, for instance. And so we're starting to know those association, and that will inform, which of the patients should not be given a particular drug, which of the patients should really be given that particular drug.
Mhm. Dr. Maytal, do you have any comments on where the field of pharmacogenomics is and your personal experience?
Yeah, sure. So in many ways, it's not quite ready for prime time on a practical kind of go to the doctor, let's get a genetic test to determine which medicines are helpful for you. There are companies, there are companies out there that do genetic testing and will generate reports to tell you which medicines are better for you. I'm speaking now exclusively regarding psychiatric medications. Personally, I'm pretty skeptical of that at this point, except in very rare cases when it is clear that something is amiss. For example, very, very tiny doses cause extraordinarily difficult side effects, or vice versa. Very large doses of medicines have no effect. At that point, it might make sense to do a genetic test to see if you're a very slow or a very rapid metabolizer of medication. But apart from those kind of very rare situations, it's not quite ready for prime time, as they say, in day-to-day clinical work, at least in psychiatry, which is what I know.
Another question for Dr. Rubin about the idea of sort of, where the field is going, where you could take a blood test and it would help you understand your risk about cancer and other non-cancer diseases. There are companies out there that will give patients scores based on like a polygenic risk score or other scores based on their DNA that's analyzed, and tell the patients, you're at this percentage increased risk for certain cancers. Now in the medical field, we're not doing that for patients. And I guess as with your background in genetics research, I'm curious how you would explain those results to patients, and how you would counsel them what to do with a result like that when we really don't have medical guidelines to go by.
I mean, obviously, I will say here, and I would say it otherwise, and I will tell my friends and my family that it's much better to do that in the clinical context, and with the guidelines and the counseling of the entire medical field. And I don't have a lot of confidence in the marketing surrounding all of these companies. Their incentives are very clear. I think that's where the field is going, but the field is going is going rapidly on the technology side and slowly because we need to be very careful, and we really need to accumulate a lot of data. And in order to really make predictions that are valid, robust, and applicable to a large patient population, we need to generate a lot more data. So that's particularly true. I mean, we talked about psychiatric disease. Very clearly, we have diagnoses that are not extremely quantitative and molecular, right? We see symptoms and we have a diagnosis based on that. But when we talk about the genetics, when we talk about the drugs, that occurs at a level that is really in the cell, at the molecular level. And that's where we need to generate a lot more data, and maybe we need to subdefine those diseases based on the molecular information, for instance. The other aspect, and I talked about it very briefly during my talk, a lot of the information we have in medicine in general, in cancer in particular, has been derived from a very small subset of the population, namely patients of European ancestry. And that's been a huge problem in medicine, that is still a huge problem in medicine. And when we make prediction based on the data we have, it is partly can have some performance on the patients that are coming from the same population, but that performance might be decreased in other population. And so if we really want to make predictions for a large number of patients, we need to generate a lot more data upstream of those predictions on a much more diverse set of patients, so that our predictions will be robust at the end of the process, right? And so I'm in the field of generating a lot of the data, helping to build a model to make predictions, and the doctors will have to assess the quality of the predictions we make in the research field and apply that to their patients one by one. And I want to make very clear that I'm talking about a process and a long-term future of the field. I am not saying here that we are ready to take any patients have a very, very specific report for their particular condition, and that will give them the perfect drugs. Nobody can do that, even the people that claim it in ads in the subway.
Thank you. I think that was a really good way to frame it for people to understand that we're going there globally, but that we're really not there yet. The scientific and medical field. Okay. Are there any other questions from the attendees? Or any other comments from our speakers? All right, Antonio.
Oh, you're on mute, Dr. Chan.
I think you're muted.
You're right. I was on mute. Thank you. Well, this was certainly great. I mean, I definitely know I learned so much specifically from each of our three speakers. The panel discussion was just really robust, and I appreciate it. So to our audience, to our participants, I really want to thank you for your time and for your attention and for joining us and your interest in this topic. We had a good number of you, which is really nice to see. I also want to thank our three speakers specifically, as well as our two moderators. It takes time, it takes effort to put this together and offer yourself and give your time. So we really do appreciate it. Sharing your expertise and your experiences, and Nyla specifically for sharing your story. It's just really so powerful and so meaningful. I wanted to share this before we leave and end the program. So I'm going to just share this handout with you here. And you'll also receive this in an email from me. So if you could see this, I put it up during the beginning of the panel earlier, but this is basically how to go about scheduling an appointment or learning more about the genetic testing, genetic counseling services at Weill Cornell and New York Presbyterian and our cancer center. So it's an easy phone number to just contact to get in touch with us to learn more. So I'll keep that up there for you to write down. But again, you'll get that from me after, maybe next week or so in an email. And along with that email, you'll get a recording of this session. So we've been recording this. It'll be up on YouTube for you to freely share and send out to your families, friends, loved ones, and also keep for yourself for reference or later viewing. And if you have any other questions that you didn't put in the chat or you didn't want to for whatever reason that you would like answered by our speakers or our panelists or moderators, just send me an email. Again, once I send that message off, you'll have my contact information, and I'll do my best to work with the folks on the Zoom here to get your questions answered for you. So with that, I think we can conclude the program for this evening. I thank you all again for joining. Be well, stay safe, and have a good evening.
Thank you. Thanks very much, Antonio.
Thank you. Bye,
Thank you
everyone.
all. Thank you very much, all.